Two other parameters about the electrical conduction of signals in neurons are the velocity at which the action potential moves down the axon and how many action potentials you can get in a unit time. So each action potential lasts for about a millisecond. Now after an action potential occurs, it takes a little bit of time for the membrane to recover, between a half a millisecond and a millisecond to recover. So that means the fastest rate at which action potentials can occur is about 500 per second. And that's true no matter what neural system we're talking about. The other parameter of action potentials is the velocity or the speed at which they travel down the axon. And that depends greatly on what kind of animal it is, because in the world of invertebrates, the velocity is very slow compared to the world of vertebrates. The speed varies from very slow in some specialized systems up to some meters per second. Meter per second is pretty fast, but if your life depends on quickly escaping, many meters per second would be very good. Vertebrates have invented something called myelin. So this is composed of a specialized cell in the nervous system, a kind of glial cell. And during development, the glial cell will wrap itself around the axon. And then another glial cell does the same thing, and many glial cells do that until the axon is covered with this coating of glial cells. The glial cells remain alive, and that's their function in life, is to park themselves on the axon for the whole life of the organism. Now there's a small gap between each glial cell. So a little tiny bit of axon is exposed between the glial cells. The neuron is clever, and what it does is it puts ion channels only where there is no glial cell. So you have this high concentration of ion channels between each glial cell. Those are called nodes. And the consequence of that is that all the ion current that flows in to make an action potential occurs at the nodes. The wrap of myelin, this wrap of glial cells, means that where there's a glial cell, there's a very good insulation layer on the axon, and the capacitance of the cell membrane is reduced. And both of those things mean that the current that comes in at the node is very quickly moved down to the next node. Very, very fast. I don't have to rebuild the action potential at every little step of the way along the axon. I just jam that current right downstream to the next node, where I open more ion channels and regenerate my action potential. So for that reason, if you have a myelinated axon, the action potential jumps from one node to the next, and it moves then much, much faster. So for vertebrates, the conduction velocity of action potentials is at least an order of magnitude higher than it is for invertebrates. And this has allowed vertebrates to become much more complex, because you can build a nervous system with many, many more neurons, hundreds and thousands and millions and billions of neurons, and fit it into a reasonable space. And the reason you can do that is because you can make axons that are much smaller in diameter and yet conduct action potentials very rapidly. And so having lots of neurons in a small space gives you the opportunity to build a much more complex brain.